The reason I put a tree on the title slide is because what I want to focus on is the ways in which reconstructing evolutionary histories, family trees, so to speak, of living organisms is a predictive tool. Okay, now, when people think of one of the great hallmarks of science, of course, is its predictability. We can make predictions based on scientific principles. But if you think about evolution, this is something that happened in the past. So you might think this is not something you would predict because it's already happened. But in fact, evolution is as predictive as any other kind of science. Now, last year we celebrated the bicentennial of Charles Darwin's birth. Last year was actually a big deal for a lot of big anniversaries. It was also the 150th anniversary of the origin of species, the 100th anniversary of the discovery of the Burgess shale fauna, and the 50th anniversary of the discovery of Zinjanthropus. I mean, it was this big sort of massive anniversary fest that we had last year. Darwin didn't invent the theory of evolution by any means. People at that time in the 1850s were already talking about descent with modification. He came up with what we now believe to be the primary mechanism that drives evolution, natural selection. It wasn't really accepted that widely in his time, but eventually it did predominate. But it was because of him that we can now actually draw conclusions about the past. Now again, one of the things that we like to hype about science is we can make predictions. We can predict solar eclipses many centuries into the future. We can take a small robotic small machine and using simple Newtonian mechanics, maybe a few little corrections from Einstein, send it all the way to a moon orbiting Neptune and get good pictures of it. I mean, we can be that precise. Predictability is one of the things that really sets, I think, science apart from many other fields of human endeavor. And yet, how would we use the past to draw predictions? That's what I want to focus on today. I want to talk about how evolutionary histories, first of all, it'll help us illuminate the past, but also how we can use them in our daily lives. Okay, this is something that actually has practical benefit. And this goes back to things that people have been noticing. This is a little bit cut off there at the end. That frog actually does have a head. This actually goes back to even before Darwin. Linnaeus, Carolus Linnaeus was one of the first to really discuss how living things seem to form internested groups. So I can take 4 randomly selected vertebrates here, and what I would notice is that these two share similarities with each other that they don't share with the others. And so they sort of form a group of their own. But the lizard also shares things with the chimp and the cat that none of these 3 shares with the frog, and that the frog also shares things with the mammals and the lizard that none of these would share with maybe a fish. Okay, this forms a hierarchy, a set of groups nested within groups. Okay, now this is an obvious pattern. Human beings can't have failed to have observed this. What Darwin did that was revolutionary was really free us to interpret these as what they are, the products of descent with modification. It's this evolutionary history that generates these groups within groups, these hierarchical sets. And these are wonderfully predictive. I'm going to give you, first of all, how do we build these phylogenetic trees? That's what we call them. A phylogeny is the evolutionary history of a group of organisms. How do we build these family trees? I'm not going to go through the methods, but the sources of information we have are legion. We can get it from molecular data. This is very common these days. We can get information from the actual genes of an organism and from its relatives, and we can compare the DNA of different species, make some assumptions about processes of evolution, and then actually come up with a set of relationships. We can also get this information from the animals themselves, from their anatomy, their morphology. Now, a lot of people will tell you that molecular and morphological data are at odds. That's actually not always the case. Usually they give you fairly similar results. There may be a few conflicts out there, but for the most part they give you fairly similar results, and we can use both to reconstruct these evolutionary histories. Now, what do I mean by these being predictive? Let me give you an example of how these can be predictive about the past. This is a tortoise. If you've ever seen a tortoise yawn, I don't know if anyone here has ever been bitten by one. I haven't, but I've seen them yawn. They don't have any teeth. Okay, just to prove that to you, here's the skull of one, and as you can see, it's edentulous, has no teeth. Okay? Tortoises, turtles never have teeth, at least in the modern world. Now, until recently, the only evidence we had for teeth in turtles was a fossil called Progonachilles from Germany, which had teeth on the roof of the mouth. It had teeth on the palate, but it didn't have teeth on the margin like most other vertebrates have. Well, let's put turtles in an evolutionary context. Here's a family tree that shows how turtles are related to crocodiles, dinosaurs, which include birds. Birds of today don't have teeth, but their ancestors obviously did. Lizards and snakes, and then mammals down here. There is some controversy as to exactly how turtles are related to other groups of reptiles. I'm just expressing that by drawing it this way. But this family tree is pretty stable as I've drawn it. What we know is that dinosaurs, crocs, lizards, mammals, for the most part, do have teeth. Okay? What we would assume is that these teeth were present in their last common ancestor. We don't necessarily know that for sure, but we would assume that because it's easier to assume that than to assume that each of these lineages independently evolved teeth. That's just a simpler assumption, and for the most part, this holds out. So we assume that teeth were present in this common ancestor, and that this feature was then passed on to all of this ancestor's descendants. Okay? Now, what that means logically is that turtles, which have no teeth, have ancestors that did. That's a natural consequence of this pattern. All we know is that somewhere along this lineage, teeth were lost. Now, this actually gives us a prediction. We can predict that we should find, if not a turtle with teeth, then something that is more closely related to turtles than to anything else with teeth, because turtles with no teeth are descended from animals that did. Pretty simple. A couple of years ago, Odontochelys, the name was clipped off, Odontochelys was discovered. Odontochelys, the name means toothed turtle. It's from China. It doesn't have a complete carapace, it doesn't have a complete shell on the back, but it does have most of the plastron, most of the shell on the bottom, but it also has dentition right around the margin. It has full teeth. So we've actually now got a turtle with teeth, which is exactly what we predicted would be found. It was also found in rocks of approximately the age we would have expected. It's a little bit older than Progonachilles. So we fulfilled these predictions with these fossils. And the examples of this are legion. Another quick example, a dolphin, whales, dolphins, don't have hind limbs. If you look at the skeleton, they've got a little bit of a remnant of their pelvis, maybe a remnant of the femur, but no actual hind limb. And yet, they're mammals. Okay, the females give milk to their young, they breathe air, they're warm-blooded. We would expect that these would have had ancestors with hind limbs, since mammals ancestrally had hind limbs, and now we've got whales with legs. We predicted they would be discovered, and they've been discovered. So, and we can make predictions about future discoveries using this. But it's not just future discoveries of the ancient past, it's also something we can use for our own benefit. Let me give you an example. This is a brown snake, Stereria decay. This is something that you might find here in Iowa. They're common. They get to be about a foot long. They like to eat slugs, earthworms, maybe little toads if they can catch them. They're colubrids, natricine colubrids. They're related to garter snakes, completely harmless, okay, absolutely harmless. This is also a brown snake, the western brown snake, Pseudonaga nucalis. This is an Australian species. It's an elapid. They get to be about 5 feet long, maybe 6 feet long. They eat small mammals and lizards. They are related to cobras and taipans. They're elapids. They're deadly venomous. Now, first of all, people will sometimes say that taxonomy is just stamp collecting. It's just semantics. Who cares? I submit that knowing the difference between one kind of brown snake and the other can be somewhat more than just semantic, especially if you're asked to handle one. The reason I'm showing you this picture, and by the way, there was a pretty thick piece of glass between me and the animal when I took the picture. I didn't just kneel down, But and it has to do with what it means to be a brown snake in Australia. There are several different snakes in Australia that are called brown snakes, and they're called that for very obvious reasons. They are snakes, and they're brown. Hence, brown snake. They're all extremely, extremely venomous. The thing is, snake venom evolves, just like any other part of an organism. And what that means is that if you get bitten by one snake, that venom is likely to be more similar to a closely related snake than to something more distantly related. Which, if you think about it, means that if you get bitten by one kind of snake, you can predict what kind of antivenom you might need. And that's important because brown snakes don't describe what we call a monophyletic group. They don't describe a natural group. These two brown snakes here are in fact related to each other, but the king brown snake is actually more closely related to this one, which doesn't look at all like a brown snake. It's kind of a black and red snake. And so if you get bit by a king brown snake and they give you a western brown snake antivenom, you'll die. If you get bitten by a king brown snake and get red-bellied black snake, that might work. So you can actually predict, and this is useful because there are several snakes here for whom no one has ever collected venom, at least not voluntarily. And no one has ever, and no one's actually tried to see how it would react to human tissue and what kind of antivenom to use. But we can actually use this predictively. You know, if we got, this one here called the bardak, we don't know exactly what its venom is like, but it's probably going to be a lot like the death adder, since that's its close relative. And so this is something we can use predictively. This is also useful in epidemiology, which I didn't have a chance to put in any examples from the swine flu epidemic or whatever that's been going through, but just a quick example. Viruses and bacteria and other pathogens also evolve. And a lot of the characteristics that go along with them themselves evolve and are related to their ancestry. So for example, if you look at, suppose I've got some sort of virus that I find and people are getting sick from it, I want to control it. If it's in this group here, if it's related to West Nile or dengue fever, trying to control ticks isn't going to do anything. Even if I've never actually found a mosquito carrying this virus, I would predict that it's going to be a mosquito-borne virus, and if I want to start controlling their populations, I'm going to start draining ponds. I'm not going to worry about spraying fields that might have ticks, at least not right away. If I find evidence that I was wrong, okay, then take care of that. But we can actually start acting before we actually know that much about the virus itself because of its relationships. And this also has impact for timing. You can actually get information about divergence timing from phylogenies. There's different ways of doing it, but you can say something about when two different lineages split in the past, which is useful if you're trying to figure out whether you need to quarantine something. Okay, if you've got a virus showing up somewhere and you wonder if you have to start quarantining procedures, and then you find out that the virus diverged from something else a long time ago, it's always been there, quarantining is not going to do any good. You might as well not even bother. A good example is bird flu, which hit a few years ago. Where and when did the bird flu that showed up in New York come from? It turns out it's related to a sample that was collected in Israel the year before. Okay, so it's a Middle Eastern, if somebody apparently imported a bird from the Middle East that had this virus, it got into New York and spread there. So now we can say when the virus got there and we can target our quarantining procedures. We don't have to, shut everything down. We know exactly where to go to prevent the virus from spreading any further. Okay, very useful thing. It has applications in conservation biology too. And here are two mammals. On your right here, that's a black-footed ferret, a severely endangered animal of the American West, very severely endangered species. People like them because they're ferrets. They're kind of cute. Next to it is a prairie dog. Prairie dogs are not that really, their populations have been declining, but I wouldn't exactly call them endangered. Black-footed ferrets really like prairie dogs. That's usually what they hang out with. We want to conserve black-footed ferrets. The problem is, and black-footed ferrets, part of what's killing them is they have a real problem with canine distemper. Canine distemper is 100% fatal to ferrets. They will always die if they get it. But if we want to conserve the black-footed ferrets, we need to conserve their habitat. We need to conserve what they want. And getting Western ranchers to conserve prairie dogs is a lost cause. Ain't going to happen. People want to preserve black-footed ferrets. People out there want to kill every prairie dog they find. Now, I personally don't agree with that. I think that, this argument that prairie dogs are destroying rangeland is a bit overblown, but nevertheless, that's the perception, and that's probably not going to change in the near future. How can phylogeny help us address this problem? Well, one thing that was discovered, and this is a paper that a friend of mine named Pamela Owen did with some colleagues. Let's look first at the phylogenetic relationships. The black-footed ferret's closest relative is an Asian species called the steppe ferret. Okay, the European polecat, that's the one that you get in pet stores. Okay, that's the pet store ferret. Minks and others are down there, but there's a black-footed ferret. Its closest relative is the steppe ferret. And as far as we can tell from several different sources of information, they last shared a common ancestor a little over 1 million years ago, sometime during the Ice Age. What's interesting, though, is if you actually look at not only the distribution of prey preference, but also the timing of that. Modern black-footed ferrets really like prairie dogs. But if you go to if you go to deposits that are older than about 750,000 years, you'll get black-footed ferret, you'll get prairie dog. You don't necessarily get them together. So that particular preference for prairie dogs seems to be a much more recent acquisition. It seems to be an acquired taste for the lineage. And if you actually look at the whole clade here, steppe ferrets don't live anywhere near a prairie dog, but they do like colonial ground squirrels, any kind of colonial ground squirrel. So what this means is that we can now start exploring perhaps other areas for reintroductions of black-footed ferrets. We don't necessarily need to constrain ourselves to prairie dogs. I'm not saying we should just necessarily start releasing black-footed ferrets everywhere, but it does give us a range of options that we can explore. This is actually something I've been involved in a little bit too, because one of the most endangered species of crocodile today is the Cuban crocodile, severely endangered. And it's really running out of habitat quickly. Today it only occurs in a very small area in southern Cuba and the Isle of Pines. But we've got subfossil Cuban crocodiles from the Bahamas and Grand Cayman Island. So we know until fairly recently it had a much broader distribution. I personally would like to see a breeding population in the Iowa River. It's not going to happen, but and the fossils don't put them there anyway. But again, we can start looking at other places to consider reintroductions. It's also, by the way, this kind of approach is also very useful in criminology. Phylogenetic systematics, the kind of stuff that my students and I do, is now admissible in United States courts of law. And it's because of this case right here. Actually, a professor of mine when I was in grad school in Austin was involved in this, although he wasn't the lead author on it. This is based on an attempted murder case. A woman in Louisiana about 20 years ago went in, she had just gotten married, and she and her husband, I guess, were planning kids, and she went in for a checkup, and she came up positive for HIV and I think Hep C, one of the hepatitis viruses. And they couldn't figure out how this happened because she had been tested the year before and she was clean. She had none of the risk factors. She wasn't promiscuous, she wasn't an IV drug user, she hadn't had a transfusion during a certain period of time before they screened blood for those sorts of things. How did she get these viruses? Then she remembered that several months earlier, her ex-boyfriend, a physician, right after a big fight when they broke up, had come in concerned for her well-being and gave her a vitamin B shot, or so he said. And then, of course, the syringe is gone, and how do you test that? So they wanted to see if maybe there was something in that syringe other than vitamin B. They went to his office. It turns out a couple of his patients who were HIV and Hep C or whichever positive did say that he had drawn blood that month. They couldn't figure out why. They didn't need it, but everything they had was circumstantial. They didn't have any direct evidence until they did phylogenetics. HIV evolves very, very rapidly, which is bad because that's part of the reason we don't have a vaccine for it. By the time you get the vaccine, it's already evolved to the point where the vaccine doesn't recognize it. But it also evolves so rapidly that by the time you're infected by one virus, fairly soon you'll have a whole family of different kinds of viruses that have descended from that ancestral virus in your system. And so in that sense, it's, I'm going to say good in quotation marks, because it really lets you trace out where a particular strain of HIV comes from. And what they did here was they showed that HIV from this woman was descended from HIV from one of this doctor's patients. So they were able to demonstrate using this that And they tested this using all kinds of different methods. They managed to get samples of the virus from all over the United States. They didn't just test this one community. And they were able to get this guy in jail on attempted murder based on this. Okay, so very predictive and a very useful thing, I think. Okay. Now, this leads me to something that I like to call the hierarchy of certainty. Okay, phylogenetics, it's this practice of reconstructing evolutionary relationships. As scientific things go, is a fairly new thing. The formal methods that we use were only really introduced starting in the 60s, and they didn't really even start taking off until the 80s and 90s. And it really has been revolutionary in several different branches of science, including my own, paleontology and systematics. But let me just show you how it's changed paleontology as a predictive tool, okay? In the past, suppose we wanted to reconstruct something that's dead. Dinosaur, T-Rex, doesn't matter. There were 3 different levels of certainty we can point to. Direct or indirect preservation. It's right in my hands. I can see it. Therefore, I'm pretty certain that it had this. But fossils are incomplete. We don't have everything. Next down would be modern analogy. I'll show you some pictures of that. And then sometimes we just make things up, speculation. And these are, go from most certain to least certain. And this is pretty much what we had. Now, direct preservation, obviously, if you're dealing with an animal with hard parts, like a vertebrate, like in this case an alligator, I can say with pretty strong confidence that this animal had a skull. It's right there. That it had a forelimb, obvious. I can say quite a lot about this because it's directly preserved. Modern analogy assumed that similar structures in different organisms, related or otherwise, was for similar function. And so in some cases, this is a reasonable assumption. Ceratopsians had these long horn cores that were covered with keratin and life on their heads, fairly similar to what you see in a modern-day rhino. Rhinos use these for all sorts of things, to intimidate each other, to chase off predators or whatever's threatening them. It's possible that they had the same function in ceratopsians. Similar appearance, possibly same function. Not quite as certain, but more so, I think, than just imagining what they might be for. Now, phylogenetics came in, and it actually gives us, I would argue, a level of certainty that's a little bit less than direct or indirect. I'll give you some examples of indirect preservation too, but somewhat better than modern analogy, because this is testable and it's predictable. So it sort of helps us by giving us another level in this hierarchy. So let me give you an example. Here's a dead animal, Tyrannosaurus rex. Suppose I want to reconstruct this animal as completely as I can. I've got a partial skeleton. That's all I have. What can I say about this animal? Well, I can go by direct preservation. I can actually look at the bones, which is all I have. I can also use indirect preservation. The thing is, bone is a living tissue. Okay, bone responds to anything that's going to happen to it. If you change your diet, if you change your workout, if you get injured, bone will record that. If you have muscles, where muscles attach to bone, usually they leave a mark on it. If you've got a nerve passing through it or a vascular structure, some sort of a blood vessel, you can see that. Your brain interacts with your skull. That'll leave marks. This is not the same as direct preservation, but in this case, this is from Sue. This is the T-Rex at the Field Museum. People hype it as being the biggest and most complete, which is unimportant in my opinion. What is important is the quality of preservation. Okay, this is the femur, one of the femora, thigh bone. And I deliberately took this picture with the light coming in at a very low angle because I want you to see these wrinkled up areas here. Each of those is a muscle attachment scar. And using phylogeny, we can actually, and again, part of it's cut off there, but we know that T-Rex is more closely related to a bird than to a crocodilian, but all 3 of these are more closely related to each other than any would be to a lizard or a turtle. We can actually take a look at the muscle attachment sites on the hip and hind limb of both an alligator and a bird. This lets us sort of reconstruct what the last common ancestor of birds and crocs had in the hip and hind limb. And we can then assume that that was passed on to all of its descendants, including T-Rex. So we can take the indirect preservation of muscle attachment sites on the hip and hind limb, combine that with phylogenetic information as to which muscles would be expected to occur in specific places on these bones, and to a certain extent reconstruct the muscles. The muscles themselves are gone. They're rotten. They disappeared a long time ago. But we can almost draw where the muscles attached. We have an idea of how big the muscles were because that's related to how big the attachment site is. We can also very often indicate where the muscles originating up on the hip, it's inserting on the femur. We can tell sort of where it's coming from. And in this way, we can say quite a lot about the animal's muscular system, much more than you might think. The muscles aren't preserved, but between indirect preservation and phylogenetic bracketing, we can say quite a lot. Another example, indirect preservation here, the brain is gone, but the brain was inside the skull. We can get what's called an endocast, which is, it's not quite a model of the brain, it's a model of the brain cavity. In some animals, the brain fills the brain cavity. That wasn't the case here, but we can actually sort of reconstruct what soft tissues were in there. We can saw it in half like Henry Fairfield Osborn did to this one, and then make a plaster mold, or we can take a skull and CT scan it, which is what we did with Sue, and reconstruct the tissues that way. Either way, we get a pretty good idea of what's going on in the brain, or at least parts of the brain. And then combine that with phylogenetic information. This is not cooperating. There we go. And this is from a paper that another friend of mine named Hans Larsen and others published several years ago. We know what the brain of a bird is like. We can just cut one open. Okay. We know what the brain of a croc is like. Again, cut one open. We can reconstruct the endocast of several different theropod dinosaurs related to birds, and we can say something about how the brain was organized in T. rex relative to a modern bird or to some other kind of extinct theropod, which lets us say a lot more about the evolution of the central nervous system in these animals. Okay, so again, we're combining indirect preservation with phylogenetic information to say more than we could say with just one or the other by themselves. And then we can even make predictions about things that aren't even indirectly preserved. They are gone, completely gone, kaput, goodbye, to dust and ashes. What about the heart? Hearts have never been found in any Mesozoic dinosaur, as far as I know. There one was reported, but that got debunked pretty quick. Most living reptiles, most snakes, lizards, and turtles, have a three-chambered heart. So the blood comes in from the body after it's delivered its oxygen, comes into this atrium. It gets pumped into a ventricle here. Ventricle then pushes it to the lungs where it picks up oxygen, back to the heart here, into this atrium, pumped into the ventricle, and then sent to the body. The problem is there's only one ventricle, which means blood coming in from the body that's gotten rid of its oxygen, or at least some of its oxygen, is mixing with blood that's just come back from the lungs to get oxygen. And some of the blood that's being sent to the body has just come back from there. Some of the blood that's being sent to the lungs has just come back from there. It works. Many thousands of species do this, but it's not very efficient. A four-chambered heart, which is what we have, but also so do crocodilians and birds. They both have a four-chambered heart. Develops differently from ours, but it's a four-chambered heart. Separates the ventricles here. So blood coming in from the body that's oxygen depleted is sent only to the lungs. Blood that's coming in from the lungs is sent only to the rest of the body. Separates them, it's much more efficient gas exchange. Okay? Which kind of heart did T. rex have? I mean, we can sort of make all kinds of suppositions based on whether it was warm or cold-blooded, but we can be more precise than that. Let's take a look at the evolutionary relationships. Birds have a four-chambered heart. Lizards, and that's the back end of a turtle, lizards and turtles, three-chambered hearts. Crocodilians, it's four-chambered. Actually, it's an optional four-chambered heart. Crocodilians have a special shunt between the aortae up here so that when they're submerged, they can redirect blood that's supposed to go to the lungs back to the body. I mean, blood that's coming back from the body still has some oxygen in it, just not as much as it did when it came back from the lungs. And they can live on that while they're submerged for quite a long time, which is why you sometimes walk on a crocodilian and it's asleep underwater. And you're kind of worried that it's died and it's just sacked out. It's living on this. So anyway, they can do that. Wish I could. A lot of things I wish I could do. I didn't evolve with a long tongue like a chameleon. Tyrannosaurs here are related to birds and crocodilians. We would again assume that the last common ancestor of birds and crocs had a 4-chambered heart, not a 3-chambered one, passed it on to its descendants, and that T. rex inherited that 4-chambered heart. Now, do we know that it had a 4-chambered heart? No. But that's our default prediction. And this method has shown to be fairly robust. Not perfect. The same method would put a full coat of fur on a whale, but nevertheless, it does seem to work for the most part. Some things we'll probably never be able to tell, like what color were they? Now, there was a paper published earlier this year arguing that some small dinosaurs seem to have orange or reddish stripes along their tail. That's based on a small animal from a small fossil from China called Sinosauropteryx. They seem to find microscopic structures preserved around them that might have been the pigment-bearing bodies. That's never been found for T. rex, so we just don't know. And I would argue that that's not something we're going to regularly be able to predict. That's a very unusual case right there. But some things we'll just never know. I mean, was it purple? I can't say it wasn't. I think it looks goofy that way, but, you know, who am I to say? But there are other things we can say. For example, I mean, we don't know what color it was, but what texture did it have? Okay? And here we have to, again, look at evolutionary relationships. Here are tyrannosaurs, birds, dromaeosaurs. These are like the velociraptors in Jurassic Park, other theropods, the plant-eating dinosaurs, crocs up there. If we look at ornithischians, for the most part, they appear to have not had feathers. They appear to have had scaly skin like this. Now, some of them might have actually had some sort of quills along their back, apparently, but in terms of the flanks, in terms of most of the body, they had scaly skin. Sauropods, the long-necked dinosaurs, same thing. Crocodilians generally do not have feathers. Okay, again, scaly skin, just like these two. But we know that birds have feathers. Somewhere between the last common ancestor of crocs and birds and the last common ancestor of living birds and some of their extinct relatives, feathers arose. And until about 20 years ago or 10 years ago, we didn't actually know where on this line feathers arose. We couldn't say. So did dromaeosaurids have feathers? Did compsognathids have feathers? We didn't know. We were pretty sure that there's skin impressions from some early theropods, from sauropods and these ornithischians. Pretty sure we can say no, at least in terms of overall body texture, that they didn't. But somewhere along this line, feathers arose, and we couldn't say where. So maybe they had plumage, maybe they didn't. We couldn't say. Then Sinosauropteryx was found. This is the one that it was involved in that paper earlier this year that seemed to suggest orange stripes. And if you look at the textures, it does seem to have kind of a blocked pattern. But this thing has what looks like what's been called dino fuzz. Okay, it has some sort of these little hollow, probably keratinous, although they're not keratinous anymore, fibers going along the back, and they probably covered the whole body. And these, we believe, are the precursors of feathers. So we have a small feathered theropod, and this is a compsognathid. This is a little dinosaur that's a little bit further down the tree than tyrannosaurs. And then more recently, we've actually got dromaeosaurids. This is Microraptor. It's a small velociraptor relative. And this thing doesn't just have fuzz, it has feathers. It has honest-to-God feathers. You can see it's got the barbs, those little fibers that come off from the rachis, which is the central axis. It had long feathers coming off of the arms and the legs, as it turns out. So we've actually now got good evidence for feathers on compsognathids, or at least things that we think gave rise to feathers, and in dromaeosaurids. What about tyrannosaurs? Now, I have seen reports of skin impressions of T. rex that don't show feathers. I haven't actually seen the fossils themselves, but our default prediction is that they were feathered animals. Could that have changed? Could they have lost the feathers? Yes. But our default prediction is that the feathers were there. Now, this fossil here, this is a cool fossil. I had a chance to look at this a few years ago. It's from China. This one fulfilled 4 different predictions at once. Okay, it's Dilong paradoxus. It's an early Cretaceous. It's about 125 million years old. It's a small relative of T-Rex. It's a Tyrannosauroid, but it's a fairly old one. Here's the skull. The skull's about like that, okay? Up until the discovery of Dylon, what we had were Tyrannosaurs. Let me move ahead here. What we had were animals like this, Tyrannosaurs, known only from basically the end of the age of dinosaurs. They first appeared 90 to 100 million years ago, and they croak at 65. But we have all these other groups, birds, dromaeosaurids, and these other groups that appear earlier in the fossil record. Now, logically, if these are descended from the same last common ancestor as these, then this lineage, too, must go back in time. That's called a ghost lineage. So we can also predict when fossils will be found. T-Rex is big. These other ones are small. We would predict that the ancestral condition here is to be small. T-Rex has basically a two-fingered hand. It actually has a remnant of the third. It's just the metacarpal. But basically, they have thumb and index finger, and that's it. Okay? All the rest of these have three-fingered hands. We would predict a three-fingered hand down here. And all of these have feathers. So we would predict, even though we have late Cretaceous, gigantic two-fingered animals of unknown pelage, it's possible that they came from animals that were older, smaller, fluffy, and three-fingered. And Dylon was all three. Okay? It did have, I don't have a good picture of the actual fossil, but it did have a third finger. I wouldn't call it tiny, but it's certainly a lot smaller than T-Rex. And it did have feathers, or at least feather precursors. They've got them carbonized around the outside of the body. So in one fossil, 4 predictions fulfilled with one blow. And since then, they've actually found some probable tyrannosaurs that are older still in the late Jurassic, which is where they should be. But again, we were able to make a bunch of predictions. So we can say quite a lot about this fossil, even if we don't have it all preserved, if we can say something about its evolutionary relationships. Very powerful tool for going beyond just the fossils themselves. Okay? Some things we'll probably never be able to tell. The media like to portray some things as debates that aren't. For example, was T-Rex a predator or a scavenger? The answer is almost certainly yes. Okay? Really, it's not a debate. I mean, most of us would think that, A, it's fundamentally not testable, and even if it was, very few animals today will only eat things that are alive or only eat things that are dead. People like to think that cats are these pure predators. A cat of 9 lives hardly counts as live prey. And I've got several pet snakes that none of them has ever had live prey. Snakes will scavenge. But was it a predator or a scavenger? That's the kind of thing we'd have a hard time telling. Okay, so some things we'll never know. What kind of sound did they make? Did they quack? Did they roar? Did they chirp? I don't know. What color were they? Some things are still left to speculation. Some things we simply can't answer. But I submit that by knowing something about its evolutionary heritage, we can say more than we could have otherwise. And using the very same methods we would use to reconstruct the internal organs of a 65-million-year-old corpse, we can also make predictions about where viruses come from, about what kinds of preventive methods we want to apply, about when infectious diseases arose. We can make all kinds of useful predictions basically using the same techniques. So it can be a thought experiment, or it can be something that actually saves lives. It can be both. And for that, we've got Dr. Darwin here to thank. And I thank you for your time.